Welcome back to our video series on evolution. In this, our fifth and final video, we're going to look at the origins of life. Now remember, everything we've looked at up to now, talking about natural selection and Darwin and speciation, we've been looking at the origin of species. That was the name of Darwin's book. But none of that tries to explain the origin of life. And this is a much tougher question with a lot fewer answers. So, what do we mean? Where does life come from? Well, you have to understand that for a long period of time, there was an idea that was well established called spontaneous generation. The idea that living things can come from non-living matter. For example, if you had your grain stored and weren't careful about it, that mice could emanate from the grain. Now, that seems like kind of a ridiculous idea to us, but at the time it was a well-established idea. It was almost as if the grain were giving rise to the mice. In another example, if you left out meat and left it out and didn't eat it or cook it right away, and certainly didn't have a way to refrigerate it, that after a while, maggots would appear. It's kind of gross, yeah. But as if the maggots were emanating from the meat, spontaneously generating. Well, let's look at this a little closer. In 1660s, an Italian scientist called Francisco Redi made this observation that if he had left some meat out and it got rotted and got maggots on it. Now, most reasonable people at this point throw the meat away, but Redi was a scientist and wanted to observe what would happen, so he kept the meat with the maggots and he observed. And what he noticed was that after a few days, the maggots would stop moving. They'd form these little pods, these little pupa. And then a few days after that, flies would appear. They would turn into flies. And they'd fly away. Now this raised some questions to Francisco Redi. He remembered something from earlier, and so he started over with a fresh piece of meat, and he noticed that flies would come and visit the meat, feeding on its juices. A few days later, maggots, and a while later, flies. And he proposed that the maggots were coming from the flies, not the meat. So he designed a quick experiment to try to test this idea. So he had left some meat out open in a jar where flies could get to it, and in one jar he sealed it tight so no flies or air could get in, and in the other one he used a mesh top so air could get in but the flies couldn't. And after a while he found maggots in this container and no maggots here and no maggots here. So it seemed to support the idea that the maggots certainly could not come from the meat, that they must have come from the flies. Well, the idea of spontaneous generation didn't go away. In fact, it was another 100 years before we're still talking about spontaneous generation, and the idea of it was even strengthened once we learned about microorganisms. So Spallanzani in 1767 took some meat broth and heated it, boiled it so we could assure that nothing was alive in it, and let it cool down, and you could take a sample of this broth that was very clear and there were no microorganisms in it. But if you let it sit out for a few days, it would turn dark, and if you took a sample out of this broth and looked at it under a microscope, it was teeming with microorganisms. It looks as if the organisms are coming from the broth. But Spallanzani suggested that they were coming from the air, not the broth, and so he did another round of experimentation where, again, he heated the broth, ended up with a clear broth, but he sealed the top of the flask. And with it sealed, you'd wait and you'd get no growth, no microorganisms. The broth would stay clear. Later, if you took that same broth and unsealed it, then you'd get this growth of microorganisms, which seemed to suggest that the organisms are coming from the air, not from the broth. Well, the criticism of this study was that with the flask sealed, that you weren't letting air get to it, and so of course nothing can grow without fresh air getting in. So while his idea or his experiment seemed to counter spontaneous generation, it wasn't enough for people to give up on this idea. And in fact, it was again another 100 years almost before we finally kind of come to the end of this story with Louis Pasteur, with some very creative flask making. He made these flasks with these long bent necks so that air could get in. You can see here that if I bring in a pen, let me grab a pen, that we could get air into this tube. So we're getting fresh air like Spallanzani's flask we're not getting, but any microorganisms that were coming in, falling in from the air, as they fell in, would get trapped down in this little crook here, a little water in here that if they did fall in, they'd get trapped. And so you get microorganisms here, but the broth would stay clear. And then later down the road, if you took this clear broth, and it would stay this way for long periods of time, forever really, and if you broke off that stem, then you would see the microbial growth. So it took about 200 years to get rid of this idea of spontaneous generation and decide that life must come from life. So with this modern understanding that life has to come from life, we still have a question. What about the beginning? There has to be a beginning. Certainly we know that there are spiritually based explanations. Many religions have creation stories that assign the creation of life to the work of God or gods. Interpretations range from a God creating each different living thing to a God or gods making possible a world in which life could arise and develop. Look, I'm not here to determine one way or the other, and I certainly don't want to question anyone's faith or debate the issue. I think we can agree that this explanation is not part of the framework of this biology course, but we can acknowledge that this view exists. What about an alternative hypothesis? What about extraterrestrial contamination? Well, that's one you might not have heard about. There is an idea that the Earth was seeded from life from outer space. In other words, we had our planet with no life, and some meteor or asteroid, as it crashed into Earth, was carrying with it, deep down in its rocks, microorganisms or microbes. This idea is called panspermia, that the Earth was seeded from another world, that small microbes or proto-DNA was delivered to the Earth from an outside source. It's kind of an interesting idea, and heck, just as likely as any other, as we can think. You think people are upset about the misconception of people coming from monkeys? Wait till they find out that they may be aliens. The idea is that these very simple microbial things could then have basically seeded and begun the evolution that we think may have happened since then. But here's the question. Could life have developed on Earth under abiotic conditions? Could life come from non-life at least once? Let's discuss what we think to know to be true. We believe that the Earth is approximately 4.5 billion years old. However, Earth back then was not a nice place. It's not some place you'd vacation. It was hot, barren, rocky, bombarded constantly with meteors. Its atmosphere was composed of nitrogen, carbon monoxide, hydrogen, and water vapor, but, importantly, no oxygen. We had hot lava and ultraviolet light, think poisonous gases and lightning. Again, not a fun place to be. But at some point as the Earth cooled, these water vapor condensed, forming rain that created the oceans that exist on Earth today. Now this is an artist's rendition of what it might have looked like, but that's what we think happened. So here's another picture. I think it's a nice, it's a very pretty picture of what the Earth might have looked like. But let's think about this. From other evidence we believe that life began on Earth 4.0 to 3.5 billion years ago. Now notice, we got a 500,000 years of wiggle room here, and a 500,000 years or to 1 billion years from the beginning of the Earth to first life. But it's the idea that it's in these early primordial seas, in this early ocean, that life may have begun through a process of chemical evolution. So let's talk through that chemical evolution. Two different scientists, Oparin and Haldane, hypothesized that it might have been possible that if Earth's early atmosphere was reducing, meaning lacking oxygen, and if there was an appropriate source of energy such as lightning or UV light, then simple organic molecules could spontaneously form under abiotic conditions in the primordial seas. Haldane went on to say that groups of these molecules, these monomers and polymers, could acquire a lipid membrane, and then further developments eventually led to the first living cells. Now there's a lot of empty holes in this story, but let's kind of look at that idea. Well, later Stanley Miller and Harold Urey devised an experiment to try to test this hypothesis, and what they tried to do was recreate what the conditions might have been like. And so they built this apparatus where they had this, they heated up water to make water vapor, and it went through this tube, and in this container they had a mixture of gases, and these are gases we believe to have been present in early Earth's atmosphere. And so you had this hot bubbling water vapor coming through here, and they had electrodes to provide electrical spark to represent lightning or to give us an energy source. And then the water would cool down and condense and come into this area, and it basically just cycled this. They did this for like a week. They just let it run with these sparks and the gases and the mixing of the water vapor. And after a while, they turned the system off and they tested the contents of this water down here. And what they found were simple organic compounds were formed. And depending on how they changed the composition of these gases, they would make slightly different versions or different types of these organic compounds. And what this showed was it's possible under the right conditions for organic compounds to form in an abiotic system. Well, simple organic compounds are a long way from having their first life, but it is the first step that we'd need. Well, later, Sidney Fox conducted some experiments to see if polymers could form from these monomers. And based on the hypothesis that on the margins of this sea, these organic molecules might have combined on hot rocks or have used clay as a polymerizing template, it would have been hard for these molecules to be formed in the water because of Le Chatelier's principle is these reactions actually produce water, which would be hard to do in a water environment. But Fox found that under the right conditions, these molecules could clump together and combine into simple polymers and even form those spheres called protobionts. And there's a significant thing here with these spheres. I want you to think about this. If I have a sphere of molecules, it basically can create for us two environments. If I have a barrier or a sphere of molecules, then I have an inside and an outside. And these two separate environments can now be different from each other. And that's a pretty big deal because we've talked about a lot of things this year about how across a barrier or between two environments things can happen, but we have to have that barrier. So let's see what else we have here. The significance of this compartmentalization was that it creates an internal and external environment, and we need that for some metabolic processes to occur. Now, other scientists hypothesize that we'd need some self-replicating molecules. These simple naked self-replicating molecules, basically a genetic molecule, probably a simple RNA rather than DNA, would be required to kind of carry on the hereditary material. And so we have some hypotheses as to how that may have happened. Well, we still don't have a lot of definitive answers here, but we know this. If we have no life and we have to get to life, a lot of things have to happen. We have to have the creation of simple organic molecules. We have to have those simple organic molecules become complex organic molecules. We need some internal versus external environment, some compartmentalization to provide some simple metabolic process. We need some self-replicating system, some genetic system, maybe it's early RNA, and we need some form of reproduction. Well, all this adds up to is one big fat question mark. We don't know for sure. We know some parts of this process are possible. And quite ironically, we have to take a leap of faith. That some primitive form of life came to be, and that over time it became similar to modern life today. But here's some questions for you. What would this early first cell look like? It was probably bacteria-like. I have some questions for you. Prokaryotic or eukaryotic, heterotrophic or autotrophic, anaerobic or aerobic. Pause the video, make your choices, but also think of a justification. What'd you choose? Well, prokaryotic over eukaryotic makes sense because prokaryotic is simpler and that's what bacteria look like, and eukaryotic cells are much more complex. Heterotrophic, there's the heterotrophic hypothesis that it's much easier to absorb nutrients from your environment to be heterotrophic than to create your own food through some autotrophic process. And anaerobic, because the early Earth's atmosphere didn't have oxygen in it. Well, at some point, being autotrophic became advantageous to make your own food, and through some maybe early photosynthetic process, we generate the oxygen-rich atmosphere that exists today. Well, with oxygen available, aerobic organisms could come to be, and with aerobic process we could become larger and more complex. Somewhere down the road we have the evolution of complex organelles, possibly through endosymbiosis, a nucleus created to make a eukaryotic cell, giving rise to the modern domains of the eukarya. Basically, we believe this is the order in which things probably happen. But again, we don't know, and we have to take a leap of faith for this. Well, that brings us to the end of our video series on evolution in our last video, The Origin of Life. If you have any questions, leave them in the comment section below, and I hope you learned something.